that he's going to be doing. And tonight here we have a special talk, just a little bit background on Bruce. Bruce has a BS degree in chemical engineering, 27 years as a research leader for Dow Chemical. He is now the director of Search for the Truth Ministries. That's his ministry, and he's got a website called searchforthetruth.net. And so you'll find a lot of good stuff on there that you want to look at. So he's got a very unique ministry. He's given creation seminars in 19 different countries. He's been doing this for a little over 20 years, I think. And he's got 11 books out, and some of his books are on the table on the back there. And he's got 700,000 books in print. A lot of them are to the countries that he and Chris and others go to speak at. And his ministry has presented at least 500 science assemblies in 6 different countries, and he got a few more in planning. So with that, I'm going to turn it over to Bruce, and we'll have time for a Q&A as well. Let's welcome him.
All right, thank you. And that was way too much about me. Let's talk about the Bible and science and truth and reality. This is only the second time I've given this lecture, and it's very, very different. What I decided to do, because I know these seminars happen on a regular basis, and you've heard lots and lots of evidence about creation, but I want to spend our time tonight looking at just 3 of the most publicized, most advertised, most talked about in every single biology book in the world, evidences that have convinced the world around us and students that evolution's an absolute fact of science. Now, one of them is a little over 10 years old, and it deals with looking at the fossils and things we find in the rocks of the earth that convince students that they have to be millions and millions of years old, tens and hundreds of millions of years old. The second evidence we're going to look at is a little over 20 years old, and it deals with the proof that things can mutate and develop completely new characteristics and abilities and slowly change one type of an animal to another. And the students get to see this, experiments that show it happens. The third one goes back over 60 years, and it's an experiment that was done in the 1950s that every student in every museum and every internet site and every book on biology and world history has shown that proves that life could have made itself. And they all sound really, really convincing and really, really good if that's all the students are ever going to hear. Now, what we're going to do is, I've called this talk, The Grand Illusion, Evolution's Hidden Deceptions. Now, an illusion is something that's done by a magician in order to divert your attention to something else and convince you something impossible is happening by hiding something that's really happening. That's an illusion. A lie, the most effective type of deception, the most effective type of lie, is something that is wrapped in truth. It is going to sound true. There's going to be lots of evidence that seems to make it like it's the truth, but hidden in there are things that actually reveal it couldn't possibly be true. And that's what's going on all around us with this area of evolution. Now, I'm going to spend the bulk of this lecture, which is going to go for probably 50 minutes easily, over half of it on something called Lenski's experiment. Now, this goes back a little over 20 years. And it's really, I'm going to click through these, it's the poster child of evolution's proof to every student who's going to go through biology, whether it's high school or all the way to a biology degree through college. It will show up on every internet site about evolution. It'll show up on every technical book about evolution. It'll show up on every biology book. It'll show up in the museums. This is this incredible experiment that showed how a bacteria, just with random reproduction and change, developed totally new abilities that weren't there. And it's going to convince people, yeah, these mechanisms of evolution work. Now, what happened was a professor, and you're going to watch a little film, so I'm not going to describe it in detail, started to breed bacteria and took the second generation of bacteria and used them to produce the third and them to produce the fourth. And every 10 generations, he did analysis to see what had changed. And it went through 75,000 generations as of a few years ago. And it's still ongoing today. Now, it actually started, I was thinking it started in 98. It actually started in 88. So this goes back coming up on 40 years ago. Okay, so we're going to look at something about 10 years old, 40 years old, and 60 years old, still promoting evolution. But here's the why I was thinking it was 20. In 2003, after 31,000 generations, one offspring of this bacteria started to use something called citrate as an alternative food source. Now, that's widely promoted as definitive evidence that mutation can cause the upward advancement of life, because all of a sudden this bacteria is doing something that it helps it that it couldn't do before. That's what students are told. Now, there were newspaper articles, science magazine articles, headlines to catch people's attention. Many of them said something along these lines. Evolution proven as bacteria evolved the ability to eat plastic. That sounds like, wow, it didn't used to be able to do that. It had a random mistake, and now it can eat something like that and it'll survive. It features, as I said, in all these books. Here's one statement about this experiment. By Jerry Cohen, an evolutionary biologist, University of Chicago, prestigious place. He thinks this experiment is a poke in the eye of those evil religious bigots, anti-evolutionists, the undercurrent of evil in our society. See, it's not people who believe in creation. It's the words that are used that reveal the bias of the person speaking. We're not anti the person who believes in evolution. We simply want the evidence to be shown that it's a belief and not a fact, and there's some problems with it. He goes on to say, the thing I like most is that this experiment shows that complex traits have evolved by a combination of random events, mutations. And that's just what creationists say can't happen. Now, just a point of correction. Those who are promoting the Bible as a source of absolute truth and reality, and they've taken time to test it, study it, understand nature, they understand science as a tool that helps us systematically understand how creation operates. It's powerful, it's useful, and if used right, it will lead you most of the time in the correct direction to find out something is true. However, it can't prove that you know for sure something is true, because you can't know that you know everything, and everything's been done right, and you've corrected all the variables, and you've seen every possibility. But what it can do is show you if something's impossible based on what we know about how creation operates. What God has done is say He has done certain things which make them supernatural, outside of natural processes. Those things are the creation of the universe out of time, space, energy, and matter. If you leave God out, you've got to explain where everything came from, but you can't start with something. You have to believe that nothing turned into everything, and that's the basis of the Big Bang. And the laws of science show us that can't be true. It's an impossible belief. You have to believe chemicals could come alive, but you can show they're so complex that could never happen, and every experiment ever done has shown it doesn't happen. It's literally impossible. You have to believe that a bacteria could turn into a human being, and that's the point of this experiment. And what you're going to see is the deception involved. Now, what I never do in any other lecture, I'm going to take 8 valuable minutes of where I'm presenting things so you can think them through, come to your own conclusions, and I'm going to show you 8 minutes of a 35-minute film where the videographers are presenting the fantastic discovery made by Lenski's experiment. And I boiled it down to the guts of the 8 minutes without distorting anything. Because I want you to sit there and I want you to think, how is the next generation being impacted? What is being told to us? How convincing is this? How logical is this? How sincere and knowledgeable and intelligent do the people seem that are presenting this? Because I want you to have empathy for the world around us. This is what your friends, your neighbors, the children, your grandchildren, the world, what we're up against as we try to convince them the Bible's true versus what you're about to see. So just listen to this experiment, because it is really interesting what this scientist did. And then I'm going to start to peel it apart and pull back the curtain to see what's being assumed and what's really behind what you're being shown. So watch this little 8-minute encapsulation of what was this experiment that's the poster child of evolution.
This is Richard Lenski.
Turn up the volume a
He started the
little.
experiment 33 years ago, and along with a team of colleagues, he has kept it going, even on weekends, ever since. In this lab are 12 flasks of live E. coli bacteria. They are the lucky few, the ones that have survived over 3 decades of evolution in a lab.
So there are 12 long-term lines.
In 1988, a single common ancestor spawned these 12 separate populations, and ever since that day, they have been growing and dividing independently. So how have they evolved? The lab environment is very different from the one the bacteria are used to. It's much simpler, there are no other organisms present, they're kept at 37 degrees Celsius, and they live in the same solution, a mixture including glucose, potassium phosphate, citrate, and a few other things. Their only carbon source is glucose, which is limited.
Above all else, consuming that glucose and converting it to offspring, replicating as fast as possible, has been essentially what we seem to be selecting for.
Every day in each flask, the bacteria divide 6 or 7 times, which increases their numbers 100-fold. Normally, we think of generations as being limited by the time that has elapsed, but in the case of these bacteria, they're limited by the resources available to them. If they had 10 times the solution, they would increase their numbers an additional 10 times. Then, almost every day for the past 33 years, this transfer process has taken place. 0.1 milliliters, or 1% of the solution from each flask, is transferred to a sterile new flask containing the same solution. It essentially dilutes the bacteria 100-fold. This gives them the space and resources they need to grow and divide again, increasing their population 100 times, before this same process is repeated the next day. And then the day after that, and the day after that, even on weekends. This process has been going on for over 3 decades. These bacteria go through 6 or 7 generations a day. So after 33 years, the bacteria in these flasks are generation 74,500. If those were human generations, it would represent 1.5 million years of hominid evolution.
So we estimate that in our bacteria, only about 1 out of maybe 100 or 1 out of 1,000 cells will have even a single mutation. So that's not very much. By contrast, in humans, it's estimated that each one of our offspring has perhaps 10, 20, 50 new mutations. So the bacteria are extremely conservative, but there are also billions of them in even a tiny flask. And so if you have a chance in 1,000 of mutating and you have 1 billion individuals, then every day in one of these flasks we might have 1 million new mutations. So that's a lot of variation on which natural selection can act. Maybe half of those mutations have no effect whatsoever on the bacteria's ability to grow and thrive in that particular environment. They might be mutations that would matter out in the great outdoors, but this is a very simple environment. Maybe those genes aren't even expressed. Another half of the mutations, speaking very, very roughly, might actually be deleterious mutations. They make the bacteria an inferior competitor. But there's maybe, out of those 1 million mutations that occur every day, maybe there's 10, maybe there's 100, maybe there's 1,000 of them that actually change something in the cell that gives the bacteria a competitive advantage over their progenitors. And those then grow over the course of that day. Then every day, 99% of the population is eliminated. This lucky 1% prevails, and if those lucky 1% include one of the guys from the previous day that was growing at 10% faster than the other guys, it has a higher probability of contributing to that next flask, the next day's flask, and in the fresh medium, and it will continue to grow faster at a 10% clip, and that compounds by an exponential process. So the mutations, are really rare when they first occur, and many of them are lost. But once they get common, if they have that competitive advantage, they'll just sweep through the population.
But how do you know that the bacteria actually have a competitive advantage? I mean, how can you tell that they're getting better suited to their environment? Well, this is where one of the unique properties of bacteria come in. They can be frozen for long periods of time and then revived.
And so they're stored in suspended animation here. So these are the racks that contain the frozen samples of the bacteria from the various generations.
Every 500 generations, roughly 75 days, they freeze a sample of each population. By freezing previous generations, Lenski and his team have a frozen fossil record.
So our samples from over 30 years ago remain perfectly viable. And so that gives us an ability to do what I like to call time travel. We can literally compare organisms that lived at different points in time. So we can compete bacteria from generation 70,000 against their ancestor.
That's right. The way they measure fitness is by competing the current generation of bacteria against older generations. It's like a strange bacteria fight club. They thaw out the old generation, mix it into a flask with the current generation, and then plate out a sample of the solution to see the relative abundances of the two populations at the start. Then they incubate the flask for a day, and then plate them out again. And the point is to compare their relative growth rates. Which generation was better able to utilize the glucose and divide faster? Because in 2003, the bacteria started doing something remarkable.
One of the 12 lineages suddenly began to consume a second carbon source, citrate, which had been present in our medium throughout the experiment. It's in the medium as what's called a chelating agent to bind metals in the medium. But E. coli, going back to its original definition as a species, is incapable of that. But one day we found one of our flasks had more turbidity. I thought we probably had a contaminant in there. Some bacterium had gotten in there that could eat the citrate and therefore had raised the turbidity. We went back into the freezer and restarted evolution. We also started checking those bacteria to see whether they really were E. coli. Yep, they were E. coli. Were they really E. coli that had come from the ancestral strain? Yep. So we started doing genetics on it. It was very clear that one of our bacteria lineages had essentially, I like to say, sort of woken up one day, eaten the glucose, and unlike any of the other lineages, discovered that there was this nice lemony dessert. And they'd begun consuming that and getting a second source of carbon and energy.
The experiment uncovered other surprising findings, like instead of the bacteria getting more numerous over time, they actually decreased in number. But each individual bacterium got larger. 6 of the 12 populations evolved hypermutability, mutation rates 100 times higher than their ancestors. But these populations subsequently acquired additional mutations that brought the mutation rate back down. I mean, it's advantageous to be able to evolve faster than others, but if the mutation rate is too high, then offspring have too many deleterious mutations.
Whereas this power law model, which says things are slowing down but never reaching an asymptote, we give it just 1/10 of our data from the past, and it projects very accurately out to 50,000 and even 60,000 generations when we last looked. It predicts sort of the future course of the evolutionary trajectory. And to me, that's kind of profound, and it's sort of changed the way I look at this experiment and even a little bit how I look at life on Earth. I mean, life on Earth doesn't stop evolving. We know that. And we know that, but we think that's because they're asteroid impacts. That's because of human impacts. That's because they're viruses that are attacking their hosts, and the coevolution is causing evolution to never stop. The world is always changing, so of course evolution never stops. And that is 100% true. But what our experiment suggests is that even in the absence of environmental change, there are so many opportunities of smaller and smaller magnitude to continue to make progress that, in fact, progress probably would never stop, even in a constant environment.
but that is a personable professor. He's friendly, interesting, enjoyable to listen to, knowledgeable. Students are just going to get sucked right in. Now, I'm going to give 3 examples. I'm going to go back and just look at a few little 10-second clips of what was said to show you this is where the sleight of hand happened. This is where deception, and he knows his deception was just thrown in. Because he's so convinced at the end, he's showing all these animals that are obviously, they've evolved. Everybody knows evolution's happened. It's a fact. And by the way, we can't be critical of people lost in that belief system. Because the only alternative to some sort of an ape having turned into a person is that God supernaturally, one day in here on earth, made 2 fully formed people. And there is no student in our country that could walk into a public university or biology class, world history class, and say, that's my belief, without being ignored, ridiculed, ostracized, disbelieved, considered a Neanderthal ignorant. The world's been trained that isn't possibly true. Well, if that's not true, the only option is that some animal turned into other animals, so it's considered a fact, because the truth's being left out. See, science works, but it also works to show us changing one totally different animal into another can't possibly happen. But what about this bacteria that changed? Well, remember Paul Harvey? Now, the rest of the story. Young people don't get that, but us old folks, it's like it was like the Rush Limbaugh of our day. All right, I'm going to just start by re-showing a few seconds of one of those little clips of what was being said, and we're going to look at it a little bit more critically, a little bit more with our brain, thinking about what's being said.
Bacteria go through 6 or 7 generations a day. So after 33 years, the bacteria in these flasks are generation 74,500. If those were human generations, it would represent 1.5 million years of hominid evolution.
Okay, so our generation's about 25 years. Those bacteria have 7 generations a day. You change the ratio, those 30 years with 74,000 generations would be like 1.5 million years for human beings. So if they're remotely similar, the amount of change in that bacteria that we saw happen should be about the sort of change we see as they show this ape being morphed into a human being. Well, that's just artistry. What you've got to do is change the information in the DNA for any of that to happen. So let's just look at this. Human and apes have similarities, without a doubt, but we're not 98% identical. We have about 3 billion letters in our DNA code, and it's a language, it's a code. Every language, every code has an intelligent designer. But now that we've decoded ape DNA, we've decoded human DNA, depending on how it's done and how it's analyzed, the difference between us and apes is not 1 or 2%. It's somewhere between 10 and 20%. Now, let's do the more conservative number. 10% of 3 billion is 300 million individual changes to turn an ape or a chimpanzee into a human being. 300 million letters have to be rearranged. 300 million mutations that have to have a positive, functional, useful purpose, or they would be lost or eliminated. All right. In 74,000 generations, one time a mutation happened that happened to give that bacteria an advantage. It could now eat an alternative food source. So in 74,000 generations, which is about 30 years, it would have happened twice. They hadn't necessarily found the other one. Okay, that's the rate at which it changes. Let's put this in perspective. So a human needs 300 million changes to explain why we're not apes. In the 1.5 million years we supposedly evolved, we should have only seen 2 happen. That's the comparison. That's like we would have seen 2 happen. That's like 0.000007% on the way from changing of an ape into a human. That's the percentage. Now let me put it in perspective. Just that something we can visualize. The moon is 239 million miles from Earth. That's 1.4 billion feet away. If you multiply 1 billion feet by that percentage, that would be 10 feet. So, in 1.5 million years, if it's the same ratio as this bacteria, a bacteria would have moved 10 feet in the direction of turning into a human being, or an ape would have moved 10 feet in the direction of turning into a human being, when you need the distance from here to the moon to explain where all that information would have come from. Just start to understand the scale of here. This is the poster child of proving apes have turned into human beings. Shows we could have, in the amount of time they proposed, moved 10 feet in that direction. See, this is not what's being portrayed by this film. I use this because the world record for how high a human being could jump is 8.1 feet. Let's round it up to 10 feet. That's the world record for a human being being able to jump, how far they can jump up. But we're being told that we should be able, if we wait long enough, to jump all the way to the moon. By proving we can jump 10 feet, does that prove we could jump to the moon in enough time? That's the comparison. Show this to the kids and then show them the scale of the change that we're talking about. And you start to understand the absurdity of believing this little change really explains how an ape could turn into a human being. It's ridiculous. It's not even remotely close. This morphing of an ape-looking figure in an artistic video to a human is fantasy. You need 300 million changes. Now, number 2 deception is this beneficial mutation rate that was mentioned. Watch this part of it.
So we estimate that in our bacteria, only about 1 out of maybe 100 or 1 out of 1,000 cells will have even a single mutation. So that's not very much. By contrast, in humans, it's estimated that each one of our offspring has perhaps 10, 20, 50 new mutations. So the bacteria are extremely conservative, but there are also billions of them in even a tiny flask. And so if you have a chance in 1,000 of mutating and you have 1 billion individuals, then every day in one of these flasks, we might have 1 million new mutations. So that's a lot of variation on which natural selection can act. Maybe half of those mutations have no effect whatsoever on the bacteria's ability to grow and thrive in that particular environment. They might be mutations that would matter out in the great outdoors, but this is a very simple environment. Maybe those genes aren't even expressed. Another half of the mutations, speaking very, very roughly, might actually be deleterious mutations. They make the bacteria an inferior competitor. But there's maybe, out of those 1 million mutations that occur every day, maybe there's 10, maybe there's 100, maybe there's 1,000 of them that actually change something in the cell that gives the bacteria a competitive advantage over their progenitors.
Okay, didn't that sound logical? Well, here's the rest of the story. Every mutation is exactly analogous to taking information, which is what the DNA code is, and randomly marking out a letter, moving a letter, rearranging a letter, sticking a word or a letter randomly into the text. Now, the rate that a random change to a book, by randomly changing letters or words, will become a more well-read, more informative, higher level of information sentence is not 0.1% of the mutations. It's not 0.01%. It's not even 0.001%. You cannot find, and this is the president of the Institute for Creation Research speaking at I believe it was Harvard University last year, said there is not a single documented scientific paper or report in any science book, in any textbook on the planet with a single example of any mutation which has added a higher level of information to a creature. Not one example ever shown. But sometimes a mutation, even if it destroys information, can give that animal an advantage. And the textbooks are filled with these, and they're filled with examples of changes in a creature that give them an advantage, or adaptations of animals. But those things are all programmed into the mechanism of the cell in the organism. They're already pre-built in, and that can be demonstrated. But like salamanders or fish can go into a cave where there's absolutely zero light, and their eyes will literally stop working. And it can be traced back to a trigger or a mutation switch or a random change in the fish. Now their brain isn't trying to process information because it can't see anyway, frees up space, so its brain is actually more efficient, and it's an advantage that it's lost its sight. But if you added information content to the animal, no, it's lost information, but it's an advantage. All right, so the actual mutation rate of useful mutations is not 0.1% or 1,000 out of 1,000,000 mutations. It's so small it's essentially unmeasurable, like 0.0001% might be useful. And he knows this. So the number he threw out was a flat-out lie. But in order to believe in evolution, you gotta just move people in the right direction. So he inflated the number enormously. Meanwhile, he said maybe 50% of the mutations have no effect, probably more like 90%, because our cells and our bodies have all sorts of abilities to have backup ways of doing things and error correction ways and backup systems. And God built all creatures with all sorts of robust systems to help them operate. So most mutations don't seem to have an effect. But what about the ones who are deleterious? Because remember, every time you move a word or a letter, you're causing some change, and many of them are going to destroy meaning. So most mutations, vast number of mutations, are neutral or they're negative to some degree. That's a fact of science, and he just glossed it over. Now, that's exactly like a car that gets a little speck of rust. That's a deleterious mutation. But it's so small you don't even notice it. But then over the next day, there's more specks, and over the next month, there's specks, and over the next year, there's specks, and there's more and more little specks. So there's 1,000,000 times more little bits and pieces of rust versus every little bit of piece of rust that you might brush against and knock off. So which one's going to win? The few or the many? The whole system's going to eventually fall apart. That's what we see happening all around us. By the way, Dr. John Sanford, he called this genetic entropy. Used to be an atheist his entire genetic career. He's one of the world's top geneticists, worked for Cornell University. He invented the ability to take genes out of the nucleus of a cell, of a plant, and then splice it into the genetic information of another plant to create better varieties. It's called the gene gun, patented, used worldwide in science labs. This man invented it. He's world-renowned. He realized every biological creature is deteriorating downward at an alarming rate. Every human generation, as he said, has 10 or more permanent, random, detrimental changes to the next generation. It's why lifetimes have dropped off from 100, 700, 800 years at the time of Adam and Eve in the next 10 generations to in the 300, 400, 500 immediately after the flood, to 200 by the time of Abraham, to 80 by the time of Moses. Because every generation has more and more and more mistakes. And modern science has allowed us to solve some things, but we continue to deteriorate. That's reality. He spoke at Michigan University about 10 years ago. I helped sponsor this conference. We brought in PhD astronomer Don DeYoung, PhD geneticist John Sanford, PhD physicist Jerry Bergman, a physiologist. He's a doctor of teaches in medical colleges. Advertised it was $10,000, invited the whole campus, 40,000 students. We challenged Lenski to a debate on genetics. Dr. John Sanford with Dr. Lenski, he absolutely refused to debate on that subject. It's very revealing. They own the microphone. They don't want to debate. They want to just present one viewpoint. So that's the situation we're in. By the way, in that conference, weekend conference, Everybody invited, professors, students, advertised at football games, advertised in campus, spent $10,000 on flyers, advertisements. 40 students came out of 40,000. Because that's how they're conditioned. Anything dealing with creation is fantasy religion. The universities are an absolute wasteland of people looking for truth. They're trained to think in only one way. So none of the reported mutations added new complex information and allowed the bacteria to survive better in a natural setting. Now we come to the last deception, then I'll move on to the next subject. So all the mutations lowered fitness. Some of the strains lost the ability to break down their food. Many of them lost ability to repair their DNA. Many of them had reduced flagellum to move around. But the lab food was so abundant that even the inferior ones survived. But this wouldn't happen if they weren't in this laboratory setting. So even all these problems, they often survived. But none of them got better, except the one at 31,000.
Which generation was better able to utilize the glucose and divide faster? Because in 2003, the bacteria started doing something remarkable.
One of the 12 lineages suddenly began to consume a second carbon source, citrate, which had been present in our medium throughout the experiment. It's in the medium as what's called a chelating agent to bind metals in the medium. But E. coli, going back to its original definition as a species, is incapable of that. But one day we found one of our flasks had more turbidity. I thought we probably had a contaminant in there. Some bacterium had gotten in there that could eat the citrate and therefore had raised the turbidity. We went back into the freezer and restarted evolution. We also started checking those bacteria to see whether they really were E. coli. Yep, they were E. coli. Were they really E. coli that had come from the ancestral strain? Yep. So we started doing genetics on it. It was very clear that one of our bacteria lineages had essentially, I like to say, sort of woken up one day, eaten the glucose, and unlike any of the other lineages, discovered that there was this nice lemony dessert. And they had begun consuming that and getting a second source of carbon and energy.
Okay, sounds really logical. New ability. It can now eat food it couldn't eat before. Mutation caused it. Case closed, evolution happens. But here's the rest of the story. It's so deceptive, it's just stunning. E. coli already has the ability to eat citrate. It already has that ability. You see, it's a harder carbon source to use. You have to have oxygen combined with the carbon to turn the carbon and break the chains and turn it into food, nutrition for the bacteria. But in low oxygen conditions, where there's not a lot of oxygen available, a switch flips inside of the bacteria, and now it will start to eat citrate instead of sucrose. Because now, without as much oxygen, the citrate becomes easier to process. God designed this bacteria to eat alternate food sources. It was already in the programming. And in the mutation, the switch broke. So now it eats both of them in an equal amount. Except the other bacteria doesn't eat the citrose because there's lots of oxygen. Well, now this bacteria, only in an artificial laboratory situation where there's a bunch of both, can eat either. It has an advantage. See, there's no new information, no new programming, no increased complexity. It's a broken switch that gave an advantage in a very artificial situation. This bacteria would not compete in a natural environment, and it would just go extinct. Isn't that interesting? That's the poster child of proof that apes have turned into human beings. It's the best they have. And it's all smoke and mirrors and fantasy and deception, because the things left out to help students come to a logical conclusion aren't talked about. The carbon source is harder to metabolize and normally not used. The switch breaks, and the bacteria would never survive in the real world. You see, there's no new information. In real worlds, they wouldn't survive. The DNA information is deteriorating, and it proves life is devolving downward, not evolving upwards. Oh, if students saw both viewpoints, the evidence, they could think for themselves what that's really showing, they'd start to search for what's the real truth about where I came from. But they're programmed to only think millions of years, billions of years, lots of time, random chance. If it's useful, it changes one animal into another. Not a single example can be shown of increased information content via mutations. There was no objections, no examples, no questions, no rebuttal to what Gary Galuza said, head of ICR at a major university, because it's true. It's one faith versus another, but the faith in the naturalistic explanation for what God said he did supernaturally isn't there. It's contradicted. See, we've got to show the people around us this stuff. Now, that took a while, but it wasn't complex, and it could be summarized in a few sentences. Now, I'm only going to spend a few minutes on the next two. I spent so much time on that to just do more of a deep drill and just help you understand how powerful these deceptions are. And it's not that hard to show what's wrong with them if we're willing to take the time to figure it out. The Urey-Miller experiment. Now, this one's kind of stuns me. This goes back 64 years, okay? And here's what it is. It was done in 1953, well, 63 years. Every biology book, every museum, every Earth history has it in there, and it supposedly shows how the building blocks of life made themselves. What's the building blocks of life? See, life is made out of chemicals. A cell is incredibly complex, and it's made of a lot of really complex parts, thousands of parts. The simplest bacteria has over 1,000 parts, and the parts are very precise, three-dimensional shapes, just like a machine. In any machine, whether it's an automobile engine, whether it's an electric fan motor, whether it's a watch, whether it's a key that fits into a lock, the parts have to be exactly the right shape or the machine doesn't work. That's exactly the same as it is with a cell. Now, how do the parts of a cell get the right shape? The most common chemical of life is called a protein. Now, tomorrow morning we're going to do two talks. One of them is called I just drew a blank. It's called the Blind to the Obvious. And it's going to look at three different examples, one from biology, one from geology, one from astrophysics, astronomy, of how we can absolutely know the Bible is true and cosmic, chemical, biological evolution can't be true. And we're going to go more deep into this, but a protein molecule is like a long chain of beads, and each bead are known as amino acids, and the amino acids are like the little building blocks, Lego blocks, that you can put together to form a structure. Now, the shape you put together with a bunch of Lego blocks can be very different. You can take like 20 different basic blocks. If you put them together a certain way, you might get an airplane. Same 20 blocks, you put them together a different way with lots of multiple of the same shape, and you might get like an automobile. You put them together a different way, it might look like a building or a house or a roadway. Probably you can have just thousands and thousands of different final structures. The final structure is like that protein molecule. It's the part inside of the machine. So God uses these amino acids, and if you line them up in just the right way, the whole chain bends and folds and forms the right shape. Are you following me? That's what a protein is. It's a little part of a molecular machine. And you got to have all the right shapes all together, or the machine won't work. But we're talking thousands of shapes, and we're talking really complex protein molecules with up to 50,000 Lego blocks, ranging from 500, that'd be the smallest protein, to 50,000. Now, let me just show you. Here's a bacteria, one of the simplest forms of life. Got to back up. Okay, right here. They found this like in a deep rock layer, which is supposedly the earliest rock layers. It's really just the first layers formed during Noah's flood. It's not older, it's just buried first. And it has 7 purple gears. Now, those gears are made up of a few proteins that force that gear to make that shape. And they have to be the right protein or you don't get the gear. Surrounded by 21 green gears, where you have a different arrangement of the protein to change the size of the gear. And they all have to be placed exactly right, so they spin these little fibers that form a propeller. But what turns the gear are 40 different proteins. Everyone has to be there or nothing works. Every protein has to have the right shape or nothing works. You remove one part of an electric motor and nothing works. And God built little electric motors at the back of each gear to turn the gears. Isn't that stunning? You put 10,000 of these motors in the width of a human hair. And if any part is missing, the motor won't work. And if any of the amino acids aren't lined up right, you can't even make the right part. All of this is statistically impossible to even make one of the proteins, let alone all of them, let alone get them in the right position. God has made it so obvious certain things are supernatural. And this is one example of millions inside of biological life. They're irreducibly complex. Unless all the parts are there, nothing works. Because God wants us to know the truth. So what did Miller do? He put methane, ammonia, nitrogen, water in a flask, added a spark of energy, like a lightning flash, let it circulate through a cold trap so he could capture these very fragile chemicals, and they wouldn't just go back through and be reheated. And he discovered there were a few of the 20 different amino acids that needed for life. God uses 20 different Lego blocks, 20 different shapes. Not 21, not 19, the same 20. You find them throughout biological life. And the way they're arranged makes the right parts, and the right parts makes the cell work, and the right cell makes the right creature. This experiment, and this is what students aren't shown, oxygen was left out. Because if oxygen's there, the chemist who did it knew you couldn't get the amino acids. And yet, as far down as we go in the rock layers of the earth, you always find oxidized rock. There is zero evidence that there's ever been an atmosphere on Earth with no oxygen. There's lots of evidence oxygen's always been around. They chose, for no reason, to leave oxygen out other than that they needed to get some amino acids. They knew how to synthesize them. He only got 6 out of the 20 needed. They've never been able to show where the other 14 came from. We just pretend they were also made. He got a random mixture of amino acids. You know how you have right-hand gloves and left-hand gloves? You can't put your left hand into the right-hand glove. It doesn't work. 100% of the time, every time any similar experiment is ever done in every laboratory throughout time, you get exactly a 50-50% mixture of right and left-handed amino acids. Life only links up into this long beaded chain, left-handed amino acids. It is impossible, they're totally identically reactively, to have a mixture of right and left-handed and end up with a protein that only has left-handed. It can't happen. Nobody's ever explained it. I think you'd win the Nobel Prize if you could explain how to make a useful biological protein with this experiment with only left-handed amino acids. Nobody's ever explained it. I think you could get the Nobel Prize if you could explain where all the amino acids came from. Nobody's ever explained it. They deceptively left out the oxygen. There's also a bunch of stuff made that caps the proteins so they won't continue to grow. And there's things that cause the protein chains to seize. And there's no cold traps in nature. And these have to be lined up in a very specific order or they make the wrong shape. And it's statistically, and statistics is science, statistically impossible means it couldn't happen even once in a 1 with 50 zeros following it tries. Like a trillion tries. Not once does it ever happen. And you can't even create one protein by random chance. All of this stuff is ignored. What this experiment actually shows is that life could not possibly have happened this way. Now think about this, Christian brothers and sisters. We live in a world and an education system where students are shown systematically that an experiment that shows this couldn't possibly explain where life came from, and every experiment ever done shows chemicals can't come alive, and even the people doing the experiments cheat by starting with biological chemicals that are only made inside of life, and they still can't get them to come alive, so we know it can't happen. They're trained to believe it has happened. Deception. It's not science, it's indoctrination in something that can't be true. And God wants us to know the truth. And we've got to use our mind to do it. Useless junk form, multiple problems unsolved, it literally proves life could never have started in this way, and it's all ignored. Now, the last one we're going to talk about tomorrow, and I think you've probably heard something about it, but we have now dug up dinosaur bones, other fossils, at least 120 reported scientific papers written where we have found undecayed fragments of protein molecules inside of fossils. In dinosaurs, we found stuff, it's so fresh you can stretch it and it will snap right back. I don't even have, tomorrow you'll see pictures of this. We found undecayed DNA, undecayed carbon-14 that's still there. All of these things have half-lives where every 5,000 to 10,000 years or less, they disappear. Half of them's gone. Proteins should be 100%. Every single amino acid should have flown off and the chain should have fallen apart. They're protected inside of a living cell. Outside of a cell, they just start to fragment and fall apart within 5 to 10,000 years. There shouldn't be anything left. We find them in fossils, hundreds of examples. Now, what's the world do to try to explain how can this fragile biological stuff still be there in fossils that have turned to solid rock? It's irrefutable that they're there. Now, we'll give lectures on dinosaur tissue and evidence for creation in schools, and I'll guarantee you the students are going to just immediately say, Google, how can fresh, soft dinosaur tissue still be around after millions of years? And up will pop the answer. Iron from the blood preserves the tissues so it can last longer. And that's what it will tell them. Now again, enormous deception going on. Now I'm going to show a little video, and by the way, let me just get to it. Came out in 2014 for the first time, and the narrative is the story to support evolution, the recovery mechanism to explain the impossible, is the idea that iron from your blood comes out, and I'll just read this from an internet source. Iron chelators increase the fossil's tissue, the scientific jargon's just over the top, immunoreactivity to multiple antibodies dramatically, suggesting a role that iron in preserving and masking the proteins in fossil tissues. In other words, iron caps it, protects it, and allows it to last for millions of years. Okay? And that's a scientific paper. On Google, it'll just say iron preserves it and makes it last longer, like a scientific fact. Now, let me show an interview I had with a man who's done a lot of work on finding this soft dinosaur tissue way back early from 2005 up to 2014 and beyond. And he looked at the work done by Dr. Mary Schweitzer that supposedly proves iron can preserve this stuff longer. So watch and listen to what he has to say as I interviewed him for one of these. By the way, this is one episode in the Rocks Cry Out series. It's an 18-part video series. It's been filmed all over the United States, in the Grand Canyon, in science museums, speaking to scientists, rocket museums, in Nashville where they have a copy of the Parthenon where I'm talking about ancient civilizations. It deals with genetics and geology and anthropology and science and biology and physics and so on. Each episode's about 15 minutes long. It took my videographer, filmer, editor person 3 months. No, it was 2 months. We did 6 a year. 2 months to create one of these 45-minute videos. So they cost me $10,000 each to produce, with the traveling and the editing and the paying of this young Christian man who did it for me. So it's a $200,000 project. Okay, it's a phenomenal series to use in a Sunday school. Lectures like this, they're about the Bible, but they're about science and reality and truth and evidence. And they just make a great thing to watch with great-grandchildren or pass on to your children or say, you'll love to do this in your church as a Sunday school class or a small group fellowship class. $40 for all of this material. This is a little piece of one of those videos. So, and by the way, it starts with a little clip from the very original Jurassic World film. Now, there were a bunch of Jurassic Park films, and there was about a 10-year gap, and in 2015, Jurassic World came out. Mary Schweitzer did her work with iron, supposedly preserving tissue, in 2014. Her paper came out. 1 year later, Jurassic World comes out. Watch this little clip the little nerdy kid is talking to his older teenage brother, and he listened carefully to what he's saying about, oh, iron comes out of the blood, and this is how dinosaur tissue is preserved. See, this isn't just scientific stuff. That movie was watched by hundreds of millions of people, programming them that, oh, some might hear about dinosaur tissue and how that shows a recent creation of the rock layers and them being buried by a worldwide flood only thousands of years ago. No. Here's the real truth. So watch this, and then there's about a 2-minute interview with this leading scientist who's been studying soft dinosaur tissue. And he's going to talk about this experiment that supposedly proves, oh, iron explains it all. Okay, here we go.
Soft tissue is preserved because the iron in the dinosaur's blood unites free radicals, and those are highly reactive. So the proteins and the cell membranes get all mixed up and act as a natural preservative. DNA can survive for millennia that way. So now, even if the amber mines dry up, they'll still have bones.
As you mentioned earlier, Dr. Mary Schweitzer kind of opened this whole door to soft tissue in specifically dinosaur bones in the early 2000s. And yet she recently published a paper saying that it was iron coming out of blood which allows the preservation of tissue, and that explains it. And it really could be millions of years old because iron's protecting it. Now, you've done some thinking and some research along those lines. What's your rebuttal to that proposal?
Right. Well, her theory is that iron and oxygen had a role. She didn't come out and say, here's the smoking gun, we found it. She said they have a role in the preservation of the tissue. Now, you have to understand that her experiment, I sort of relate to a 10th or 11th grade high school experiment, because she took soft tissue from chickens and ostriches, and she took blood, and she highly purified the blood. In other words, she used a whole set of ultracentrifuges to spin everything down and to take out, blood clots very quickly. There are all these factors in the blood serum and in the cells, in the tissues, that cause blood to clot quickly. It's a wonderful mechanism of keeping our bodies intact.
Sure. And the iron is tied up in the blood
Correct.
cells.
The iron heme is from the hemoglobin, and it's tied up in the red blood cells. So she had to remove every single cell, white cells, platelets, subplatelets, and all the serum, because it had all the clotting factors in there. She had to pull all that out. And she had to use an anticoagulant. She used a chemical that prevented the blood from clotting after she took it out. Then she took all the cells and everything out. Then she burst open the red blood cells and again used ultracentrifuges to spin all this down. Now, when I went to Hell Creek, I didn't see any ultracentrifuges. I didn't see any anticoagulant anywhere. I just saw bones in dirt. And so the other thing she did is that she put all this in a bucket. I say a bucket, a container on a laboratory bench in an air-conditioned laboratory for 2 years. It was not exposed to water or the freeze-thaw cycle or the heat of the Montana summers or the insects or the microbes or the bacteria or the plant roots. The horn that I found was loaded with all this stuff.
Wow.
And so 2 years on a laboratory bench does not a 68 million year explanation make.
So, in summary, she used totally unrealistic, not related to a natural world conditions to try to justify a belief.
Correct. And she had to, because she painted herself in a corner, Bruce. She proved beyond any shadow of a doubt that this is original dinosaur protein, original dinosaur cells. She nailed the coffin shut on that. Now all of a sudden she's got to make some kind of an explanation for how this stuff is still here. Because again, the pet we bury in the backyard, we know breaks down and
it's not there.
Goes to nothing in years.
10 years, let alone 100,000 or a million years.
Correct.
Yeah.
Okay, so you get the feel for it. Enormous problems. What kids are being taught isn't true because all the issues with it are being left out. They're not being trained to think critically. What's the truth? Not what's the possibility that might help me hold on to my belief system. So the collagen in our skin is a protein. It's 97% collagen. That's the kind of stuff we're talking about. And it's still soft and stretchy and snaps right back inside of these dinosaur bones. It can't be there. The lab reports show not a single molecule of collagen can remain after 30,000 years. Brian Thomas from Institute for Creation Research, he's recently found as many as 120 scientific articles showing there's still undecayed proteins inside of solid rock fossils that are supposedly 300 million years old. She's maybe, with absolutely impractical methods, shown how maybe you could make a protein instead of lasting 30,000 years, maybe you can get it to last 300,000 years. Well, that's 10,000 times shorter than 300 million. It doesn't even begin to show how it could still be there. So in summary, the information students receive is highly managed. Evolution's assumed to be a fact, and it would be deceptive to show them anything that contradicted evolution. Whatever fits the narrative is shown. Whatever contradicts the narrative is ignored or explained away in ways that aren't reasonable. Education is not looking for truth in our system, because by law, separating church and state, church deals with God, state deals with something that has to not have God. You can't consider the possibility that he's intervened or interacted. So intelligent people, and tomorrow, if you've got an hour, come to the why. Are people like Richard Lenski so blinded? Because I don't think he's out to deceive. He himself is blind to these things. And how can that be? And then what do we do about it? Expose the deception. We got to. It's what these resources are all about. Point people back to God's Word. And here I just talk about the whole progression that God's anger, his wrath, is revealed against those who suppress the truth. Why? Because they do it in unrighteousness. They want to get out of accountability from being under God's judgment. And what better way than that to ignore he exists? And what better way to do that than to ignore that creation has a creator? And the response to that are the three things in that first chapter of Romans, whereby God, when we choose to ignore the truth, which is obvious, God will give us up to uncleanliness and the lusts of our heart. And if we continue to choose to ignore the obvious, that God is creator, God will give us up to vile affections. And we continue to choose to ignore God's existence and his holy justice, God will give us over to a reprobate mind. And then there's that laundry list of issues that will invade us culture. You know, hatred and war and anger and deception and just a laundry list. And that's what we see all around us. Now, I want to take 5 minutes, and we'll still have time for questions, because if I don't mention the resources, you don't understand the value. Now, for not just for 20 years, I've been speaking, teaching, and writing on creation for over 35 years. And I discovered early on, people don't need to be turned into little technical scientists and nerds. And they're not interested in that. And they don't want a bunch of technical details, and they're busy, and they're boring. But the wonder of God's creation, everybody can see. So all this stuff I've produced is really visual, really short, and really pertinent and relevant. Starting with the devotionals, if you've never seen one, every day of the year, there's a short little article that's like really, really well illustrated, dealing with some fish that can see things outside of the water and grab birds out of the air because it can see it. Or the complexity of babies growing inside of us, the articles on anatomy and fossils and biology and genetics and just stuff from science, but on a fun, interesting level with 2 or 3 paragraphs and a Bible verse. These books are given away by the dozens. Everything on my table is 3 books for $25, so it's like $8. They're even lower when you buy them in bulk to give them away, everything I produce. I just produced a book called A Christian's Guide to Refuting Evolution. Everything I just talked about is there on a couple pages dealing with chemical evolution and biological evolution. But it deals with all the examples in the textbooks, all the examples in the museums, and it's filled with activities you can do with children to talk about, well, what good is half of an animal or a part of a feature? And it's got all sorts of QR codes to watch videos like this and other videos. It's just a great tool for anybody who wants to dig a little deeper. If we're going to reach the children, we've got to do it with fun, interesting stuff. Tracy Lowe, my assistant director's wife, and I just finished a book, and it's like the Chronicles of Narnia kind of book, a book adults absolutely love to read, but it's aimed at 6 to 12-year-olds because it's got this phenomenal narrative about an atheist professor who's like more brilliant than Einstein. The IQ charts can't even measure his IQ. And he's got all these fantastic inventions, and he's done everything and predicted future trends, and he's a trillionaire, and he's had famous jobs, and he's done everything, but he's not content because none of it's brought contentment. And the two orphans show up at his house. It's got a narrative that runs through the whole thing, but he takes him to see all these phenomenal things of creation, ultimately realizes God has to exist, ultimately becomes a Christian, and it's just a phenomenal book. It's like fun to read. And it puts all this kind of stuff at a child's level. So not just the children, but the parents will start to see the evidence that supports a biblical viewpoint. Phenomenal book to give away to others. Every family and every church. What a gift it would be to give them this book. Now, I only brought advanced copies. Next week, I have 10,000 copies arriving. Sign up on the newsletter, and I will send you a copy as soon as I get home next week, the week after Thanksgiving. And then think about giving them away as Christmas presents to impact people's thinking. It's just a beautiful resource. The last one, other than the flash drive, I'm going to take a little over 1 minute because this is like a diamond that you just don't understand if you just see this teeny little book on my table. Got a book from an author in New Brunswick, Canada, about it 2 years ago. Threw it on my shelf desk. A few months later, I had some time, and I pulled it out, and I read one chapter. Now, a chapter is literally 2 pages, a page in italics and another page. And there's 50 different little stories. He calls them reflections. I've never, in 35 years of teaching creation, seen a book like this because it's aimed at someone with no interest in creation, no interest in the Bible, no interest in the belief in God, but he's going through life and he's been trained to think about everything having been made by itself. So he doesn't even get to the authority of the Bible or the idea that it might be God's word until way at the very end of the book. But these are like some of the chapters. This is a chapter about design. Okay, so the author is sitting in a beautiful meadow on a summer day, and there's italics for the first page. And then there's non-italics where he kind of zings in on the point. And by the way, his writing was entered individual short little articles as short stories in a writing contest in Canada. In 2018 and 2019, he kept sending in his little stories. These stories won first place in an entire province in Canada and second place both in 2018 and 2019 as the best stories written by an entire province. So that's the quality of this stuff. And I'm going to read one to you. Because how long has it been since you've been read to out loud? I bet it's been a while. 1 minute. Okay, so he's sitting and looking as the sun's coming up in a meadow. Okay, this one's called Design. First blush blends with the blackness of space. Morning swell creeps imperceptibly until the burgeoning hues descending burst and condense into that iridescent arc to the fingernail moon of a sun set to rise. The mackerel sky, salmon streams from pink to rose. It floods the dawn with a saccharine glow. Drooping like taffy, fog fingers steal off to fill empty pockets. Leaves sopped in water weight bow in forced worship. Cobwebs in a crosswind are illuminated now like lines of 10,000 street lamps as dewdrops engorged with the sun. Okay, what's that all about? He's just looking at what he's seeing out there in front of him. We blind ourselves. Go stand in the middle of your living room. Close your eyes, spin around 3 times, stretch out a finger, open your eyes. No matter what object you're pointing at, it was designed and built. No exceptions. The rocking chair, designed and built. The coffee table, designed and built. The desk, lamp, remote control, each one designed and built. It made a journey from the head to a skillful pair of hands, head to handiwork, thought to wrought. Any object you care to point at. Now, go back to where you're spinning. This time, point through the window. Point to a tree, to the ocean. Point to a turtle, a bird. Well, strange, huh? It's funny, isn't it? How so glibly we divert our thought stream. How we let our rationale take us only so far. Suppressing what so readily flows from thought. Refusing to acknowledge what's so clearly seen. Maybe funny isn't the best word to be used there. That's it. And it's just a jewel. And it's just reflections. And he talks about, going on a journey, and let's just think about what we see around us. And he deals with DNA and carbon-14 and radiometric dating and rock layers and simple cells and mutations and natural selection and all this stuff dealing with evolution. But that's how he deals with it. So this is a great book to give away. And the reason I take the time is because if you don't know that, I gave it a new title, and he allowed me to publish it, and I offered to send him back $1 a book, and he said, I don't know what I'd even do with the money, just use it to support your ministry. So again, we price them really cheap in bulk, but everything on the table is 3 for $25, and I called it Science Shadowlands, the light for the pathway home. So, all right, so those are resources. We got some time for questions. Any questions?
Yeah, we have time for about 2 or 3 questions.
And the resources are like putting a pebble in someone's shoe. Sign up for the newsletter. Every few weeks you'll get a little 3-minute video that you can pass on to somebody, the creation devotionals, the unique resources. They're like filling the shoe with rocks eventually. And just get the information in front of people. Okay.
Yeah.
So, questions?
Yeah, so we have time for just 2 or 3 questions. We don't have a roving mic, so speak loudly. Put up your hand if you have a question, and ask the question, and who's going to be the first one?
The brave first person. And I love questions because it gets at why didn't you mention this or whatever?
The experiment, they start with bacteria.
Yes.
How do they get to the bacteria?
Well, and that's a good point. And tomorrow morning when we talk about blind to the obvious, we're going to spend about a third of the lecture talking about could life make itself? Could chemicals come together? The assumption is, this is an assumption, this is the belief. It's not science versus religion, it's one belief versus another belief. The belief is, because we can make a few amino acids, the pieces of a protein, the proteins made themselves, never been shown how you get the right proteins. If you get the proteins, maybe they can get surrounded by a membrane. That's never been shown how it could happen, but that's assumed to have happened. And then given enough time, maybe it could come alive and make copies of itself. That's never been shown how that could happen. It's all assumption. Because you understand the alternative is God literally made it. And that's not scientific. Well, admittedly, because God told us it's not. He said, study, take dominion over creation, we can study, understand, and control it so we could know certain things are possible and certain things aren't, and we would know he exists. The bigger question is, did anything new develop? We started out with bacteria. What did we have at the end? Bacteria. That's what we find. Creatures vary, they adapt, they have different changes, but one body structure never changes into a completely different body structure. 10 times God said in Genesis, creatures reproduce after their own kind. Birds make birds, fish make fish, trees make trees, people make people. 10 times. That's literally what we see. It's not that there's only 10, it's that there's basic body structures. because the programming is in an animal to be that kind of animal. God's genius and creativity allows that animal to adapt and have lots of variety, but it's still that same basic body structure.
Okay, another question? Yeah, back there.
In the last couple of years or so, have you seen more openness to the information?
To the kind of stuff we're talking about? No. Much more closed. Every decade is there more openness to the kind of stuff we're talking about here? An indication of that is 10 years ago, $10,000, brought in PhD scientists, world-renowned scientists, advertised it across the board, offered to debate, 40,000 students, and 40 show up. That's not openness. That is an increasing indoctrination that leads to every generation being less interested in it. Is there more openness? Students polled now coming out of American universities, do you plan to attend church or become a member of, or deal with Christianity or whatever? Only 14% say they have any interest in the Bible or being involved with any sort of Christianity or a church. 14%. Lowest time in all of American history. When asked why, the prevalent answer is we don't believe what the church teaches. So no. The 3 generations down, God being left out of the university system, assumed evolution in every form is fact, cosmic, biological, chemical, geological, it's all fact. You're going to program even more the next generation, it's being taught even more strongly. Our churches are emptying. Seminars like this is a huge crowd. Typically, we'll get 12 people out of a church of 800. Because Christians say, oh, I know there's a creator, but they've blended in their mind the millions and billions of years in evolutionary concepts. They're still Christians, but they're ignoring the straight, clear forward statements of the Bible because they've been trained they can't be true. And those who aren't Christians have absolutely no interest in it because they know it's not true. So no, it's not getting better. And that's, I think, primarily because people haven't taken this issue seriously and they haven't studied it.
Okay, I think we'll end with that question today. We will have more time for a Q&A tomorrow because remember we're going to do 2 different lectures. I think one by yourself.
Yep, one by myself, one by Chris. One of them is how can intelligent, sincere, smart people be blind to what is really obvious, and God says it's obvious. The other one we're going to talk about designs from observing things God has made that have led to inventions that have changed all of human history. And it's just a fun lecture.
So two very good lectures, and then we'll have a break so people can look at the resource tables and also refreshments. And at the end, we'll have time for a lot more Q&A. So think of some hard questions for Bruce and Chris tomorrow, and be ready to participate in that. Let me just remind you, that we have the mini conference, of course, but then Saturday night we have the youth night out, and it would have been at this church, but the church has another property which is only 4 miles away. The address is on here, 14715 McElroy Road, just off of Highway 9. So it's pretty easy to find. There are flyers on the back table that you can use to get there as well. Okay, thank you. Okay, and then just to looking at next year, the first speaker is Patrick Neary. He's been here quite a few times, and he is, of course, he's a geologist. He does a lot of training of homeschool kids with his geology. You've heard about, he's called the Rock Man. Well, next year he's going to do a lecture that you see here, which is on gemstones, crystals, and agates, okay, which is a little different. And he's going to look at those from the perspective of the flood, because these things don't form naturally today. So how did they form possibly through the flood? So he's going to cover that topic. He has founded Northwest Treasuries, and he's got another a group that does the Learning Center. And so that's where he does his lectures. I believe he does a lot of them over Zoom, so they're accessible to people anywhere. And he leads tours of Mount St. Helens and Yellowstone as well during the summer. So be mindful of those. Okay, so in the back there's refreshments, of course, and there's resource tables to look at. We have a whole bunch of DVDs on the second row of tables there, and we have a special offer on buy one, second one's free. Okay, that's about 100 different DVDs. Take a look at those and see which ones are of interest to you. If you're not signed up on our mailing list, there's a sign-up sheet on the back table, and also there are lots of different flyers there as well of the events coming up. So take advantage of that. And remember, look at Bruce Malone's books, and that he and Chris have there. And let's see, what else? Well, that's about it for this year. I'm going to ask Pastor Rick to come up. Of course, for those you don't know, Rick is the pastor of this church, and he's also on the board of the Apologetics Forum, and we appreciate him making this church available for the Apologetics Forum. And so I want people to meet him if you haven't met him already. So, Rick?
Thanks for being adaptable to our setting here, and thank you, Bruce, for your presentation. Looking forward to tomorrow. Oh, yeah, I was just mentioning donation cans are off to the sides there for those that desire to support the ministry as well. Let's just pray. Father, you are so amazing, and we've been seeing some of that tonight here and look forward to more tomorrow. Thank you for Bruce. Continue to bless that ministry. Thank you for just the opportunity to gather together in this way and have your truth set before us. Strengthen our faith, bless our fellowship, and our homeward way tonight. All in Jesus'